All right, thanks for joining us, everyone. I see participants loading in. This is our May webinar from the Soil Food Web School, Fungi in the Tropics, and we have Kennan Mighell here to show a lot of really cool pictures and tell a lot of stories about fungi that he's encountered during his adventures. So we'll just give another moment here for participants to join us in the room. As always, we're going to ask that in chat you sort of introduce yourself by telling us where you're joining us from today. We just love to see the global nature of these webinars, and we're excited to, there's already people know what to do. They're already joining or explaining that they're coming in from all over the world.
Yep.
I saw South America, Central America, I saw Europe, all over North America, Turkey,
Manchester, UK, Brazil.
Excellent. So we always start with this slide just to orient everybody who's joining us today to sort of the attendee guidelines. If you're Zooming with us today, you're going to be muted for the duration, but we do want to hear from you. So the chat is an opportunity for you to give, to talk with each other, or I'll be monitoring the chat, maybe some of the other panelists as well, and we can interact there. If you have a specific question that you'd like to ask for the Q&A session that we'll have at the end, the last hour or so of our time today will be a Q&A with the panelists. Then there is on your Zoom navigator bar specifically a spot for you to click on and ask that question. And so that will go into the bank of questions, and we'll get to as many as we can today. But just feel free to interact with each other and with us in the chat as well. And then we'll get to as many questions as we can. Now, I do want to give you just the outline of today's session. This is fungal diversity in tropical ecosystems. We're in the introduction right now, and then we'll have Kennan Mighell presenting about several of the really cool, kind of unknown to modern science species that he got to learn about on his travels. And then we'll go into that Q&A panel after his presentation. So we expect the entire thing will take 2 hours. And then here we are with just quick introductions of the different panelists. And we'll start with you, Elaine, and then go down in this order. And everybody can just give us our quick elevator length introduction to who you are.
Okay, I'm Dr. Elaine Ingham. I've been working in the field of soil microbiology for something like 45 years. So soil microbiologist, a founder of the Soil Food Web School, as well as Soil Food Web Incorporated, where we do research projects, whereas in the school we're training people about how to determine biomass numbers of all of the soil food web organisms that you need to have in your soil in order to make natural nutrient cycling, etc., actually occur. So spent a lot of time in Oregon, but off on a lot of globetrotting coming up this year.
Thank you, Elaine. And then Kennan, you're up.
Hi, everybody. My name is Kennan, and I'm a mycologist. I'm an artist. I wear a lot of different mushroom hats. Most of the hats that I wear are mushroom shaped, and I'll get into those during the presentation. So great to see all these names popping up from all over the world.
Excellent. And Laura, would you please, I think this is your debut on our official webinar series.
Yes, it is very excited to be here today. As I am from Costa Rica, so I was born here, raised here, educated here. So it's a pleasure to be part of this webinar dedicated to this region. And I am a biologist, and I'm very interested in insects, but my study in insects I ended up working a little bit with fungi because they are very related and they have like very interesting interactions. So I ended up working a little bit with fungi. And then as a part of this soil food web community, I've been working for Soil Food Web for about 2 years now. I have learned a lot of this group, and I'm still learning a lot from this group and the relevance in soil. And yeah, I'm happy to be here, and I hope that some of my students are present here today. So it's exciting.
Excellent. Thank you so much, Laura. And yeah, we can send the link to your students afterwards. Maybe they can get some bonus points with you for chatting with you about this session. So I'm Dr. Adam Cobbs. You have seen me a lot of times. I don't play the role of host on these webinars, but I'm always happy to step in with opportunities like this because we had the chance to meet Kennan, and we're really excited to bring him in here and facilitate this. I do a lot of content creation as well as science communication generally across activities here at the Soil Food Web School. And I have also been here about 20 months. You started just a little bit before me, Laura. But it's exciting to be part of this community. So I want to actually stop screen sharing here and turn this over to Kennan because he's going to drive for about 55 minutes.
Great. So let me get set up real quick. There we go. Everybody see that okay?
Looks perfect.
Awesome. Okay. So hi, everybody. Thank you so much for tuning in. Like I said, really exciting to see you all checking in from your various locations across the world. It seems like a very global group that's assembled here, which is really exciting for me. I live in Portland, Oregon currently, and I'll be talking today about some research that I did abroad in Guyana in South America and Cameroon in Africa. So I just want to preface my presentation by saying that one thing I hope that you can take away from this presentation is that you can do similar work to what I did fairly easily from probably wherever you are in the world. I saw a lot of names popping up from tropical locations. The world of tropical mycology is exploding. There's so much that we don't know. There's so many species left to discover. And if you look at your screens, you'll see this mushroom here is called Amanita robusta. And this mushroom is one of my favorite mushrooms. It's one that I worked on, and I'll talk about that in a bit. But this mushroom was described to the scientific community back in the 1920s, and then pretty much was not encountered again until my collaborators and I went to Cameroon and found it again. So even a big, beautiful, bright, charismatic mushroom like this might be hiding somewhere in the jungle waiting to be formally described. So I hope that you feel inspired to go out and explore. Let's see here, make sure that I can, there we go. So let me introduce myself real quick. My name is Kennan Mighell. I grew up in Washington State, Western Washington. I spent a lot of my youth wandering around the forest and getting really excited about being outside. It was just always kind of a natural passion for me. I certainly come from a place of privilege, which I would like to acknowledge, because that privilege was the platform from which I jumped off into the life that I've come to lead. It's opened a lot of doors for me and made a lot of these things possible that I do. I was fortunate enough to have parents who had the means to support and encourage a kid who was just naturally drawn to wild spaces. And yeah, I just want to point out, not everybody has that opportunity, and I was very fortunate. And yeah, so I hope that in the future, one of my goals is to help the wilderness become a little bit less of a privileged space to access, because there's a lot of cool stuff out there, and I think everybody should have the opportunity to get out there and explore. So yeah, I got a bachelor's degree from Whitman College in biology. During the summers of which of my university days, I was a wilderness guide through a YMCA camp. So I spent a lot of time in the wilderness on my own, sort of guiding groups of friends, but also groups of teenagers through the wilderness and having transformational experiences in a wilderness setting. It was a really cool job. I went on from there to get a master's degree in biology from Humboldt State University, now Cal Poly Humboldt. And that is the work that I'll be talking about today. So these days, like I said, I'm in Portland, Oregon. I spend a lot of time cultivating fungi, foraging for fungi, wildcrafting, and teaching people about mushrooms in whatever way I can. And so my graduate research was on, more specifically, mushroom systematics and biogeography of the Guiana Shield and Central Africa. So I'll get into those details here in a minute, but I wanted to draw your attention to this image, which I think is a pretty spectacular one. This is an image that I took out of the window of a plane. Yeah, you can see another image on the side there. So these are two images that I took from the window of a bush plane that was taking us deep into the wilderness of Guyana, where we conducted our field research. So the big background picture that you see there, I think of this landscape as kind of reminiscent of the North American Southwest, so sort of the canyonlands with the buttes and spires, canyons. So it's like if you imagine that draped in a carpet of tropical rainforest, that's kind of what this landscape looks like from above. And then on the side there, you can see a view of the mining that occurs in that region. So much of this, to get to our field site in Guyana, we would take a bush plane from the capital of Georgetown deep into the interior, and over much of the distance of that journey, you're passing over continuous tropical rainforest, and then you would see these big mines scarring the landscape here and there. There's a lot of mining that occurs there. And yeah, so the graduate program that I pursued allowed me to sort of combine my passions for mushrooms and my passion for wild spaces. And I'm very fortunate to have been able to explore this stuff. And so let me get into my project here. This is kind of a weird image. I just had to include it because it's one of the coolest things that I came across in my travels. It has nothing to do with mushrooms, but these two little guys down on the bottom right of your screen, I found in a museum in Cameroon. And they're artifacts from a tribe, I don't remember what part of Cameroon, that used these as divination tools. And then you can see in the background of this slide is what is transcribed on the top of one of those figurines' heads. And so these were divination tools supposedly used to communicate with beings from other worlds up in the stars. That is what the curator of the museum told me. So really cool, because they look a lot like aliens, right? And fun things that you come across when you visit museums. So my projects involved the genus Amanita specifically. I was sort of the Amanita guy. I would focus on those mushrooms. Many of you probably are familiar with Amanita. We'll talk about those in a bit. So this is what I ended up doing for my graduate research. I formally described 3 Amanita species from Guyana and 3 from Cameroon. I epitypified 4 species, and so that's including that pink one that you saw on the first slide, and I'll talk about the process of epitypification in a bit. Generated DNA sequence data for all of these collections that I made. I made many collections other than the ones that I formally worked on. And then this work contributed to a much larger project that was focused on biogeography, which again, I'll touch on in a minute. So I mentioned that bush plane flight where I took those pictures. Let me show you what that looks like landing here. So I'm going to show you this quick video. So this is the airplane coming into the landing strip. And again, this is deep in the interior of Guyana. So you can see the plane coming down and then it kind of disappears. And you're like, uh-oh. But it's okay, they landed. So this is the plane coming to pick us up at the end of one of our research trips. These pilots are amazing, by the way. They just treat their job with such casual effortlessness. But if you think about what they're doing, you know, they're cutting down low over the canopy of the jungle and then landing on these rough, uneven airstrips. So this was how we got into our Guyana field site. Our field site in Cameroon was one that we drove, hiked, and then canoed to. But in both cases, we were deep in the wilderness. And in Guyana, we had a group of field assistants that you can see some images of here down. So the only people that are out there in these field sites, so we take this bush plane to get there. It's about an 1.5-hour flight from Georgetown, basically headed more or less straight south. The field site itself is near the corner of where Guyana, Venezuela, and Brazil meet. And it's this region called the Pacaraima Mountains. And the only folks out there, by and large, are members of this tribe, the Patamona tribe, and a handful of miners. So when we landed at our field site, it would have been coordinated in advance. They have radios, essentially, which is where Terry was my graduate advisor. He's the guy saluting in the top right there. Mad respect for Terry. He has, he's the reason why all of this work has occurred. He's been studying this stuff for decades. If you haven't heard of him, look up Terry Henkel. He's a great guy and has done a lot of cool research along the lines of what I'm talking about. So he coordinated all this stuff in advance. He'd radio out to these guys. They'd, run out to the airstrip and meet us and help us carry all of our gear to our field site, which you can see in the background here, and you can see some timbers lashed together. So that's the frame of our field site. So they would, build these basic timber frames, and then we'd cover them with tarps and set up a little basic field camp, and we'd hang our hammocks from those frames too, and that's where we'd sleep. And I'll have a picture of that in a minute, but a couple things to point out. In the bottom right here, you can see them being very loyal field assistants, helping, because we're in a rainforest. We go to these spots during the rainy season, of course, when you're more likely to find more mushrooms. And so sometimes you've got to get that natural light for the good photo of the mushroom, but it's kind of raining. And so sometimes you need help keeping the rain off of your camera. And so that's what you can see going on there. So while we were out in the jungle, these guys, the Patamona, they were our guides. They were often our cooks. They were helping us conduct research, helping us sample the plots that were set up. And in their downtime, they were doing all sorts of interesting things too, weaving baskets, which you can see occurring in the top left there. Hunting, they hunt with a lot of the time with slingshots. Actually, they're really amazing hunters with slingshots. They would have one rifle, and sometimes would bring back bushmeat, because these folks essentially live off of bushmeat and cassava, which they cultivate through slash and burn agricultural practices. And so that's kind of where they get their starch, and then they get protein from the bushmeat that they hunt in the forest. And so they were basically out there just kind of living their lives and also helping support us. They're really, each one of these people that helped us out were just amazing, powerful, capable, brilliant humans. I have so much respect for them, and they're the ones that make this work possible. So these are some of the Patamona faces from Guyana. And here are a few of the faces from Cameroon. So in Cameroon, we were, I would say, almost nearly as remote as our location in Guyana. We didn't take a bush plane to get out there, but it was a very long drive. And then we got to a little settlement at the edge of the Jaw River, and then we would get a canoe to ferry us and all of our gear across, hike through the jungle for a little ways, and then get to our field site. So that work occurred in the Jaw Biosphere Preserve in Cameroon, and the Jaw is a protected wilderness. It's an ape sanctuary, so there are gorillas, chimpanzees, and of course a lot of mushrooms. And you have to get a permit, to access the wilderness. Almost anybody except for the Baka, who you can see here on the top left here. This is a Baka family group. About half of those folks that you see in that picture were our actual field assistants, and the rest were members of their family that would come out and visit us sometimes. We were in Cameroon in the field for about 44 days for each of our trips. So we were camped out in the jungle near these Baka settlements where these folks' families lived, and they're cultivating all sorts of food that they would sometimes bring to us. So that was kind of where we got our groceries. These Baka folks coming back and forth and saying hi to their family, and selling us some plantains and whatnot. So yeah, in the top center there you can see Mei Lin, who is an integral part of the team. On the left and on the right, Francine, who was our cook, the most amazing cook. And then those are all wild chanterelle mushrooms. So in particular, our Cameroon field site was loaded with edible chanterelles. So we ate a lot of wild mushrooms on these trips. On the bottom left here, you can see a couple of local students who were helping us out, helping us measure spores and that kind of thing. And then that image there in the background in the center is one of the huts that the Baka build, and they're sort of semi-nomadic, so they roam around the forest, they build these huts, live in the space for a while, and then they kind of move on. So yeah, again, these field assistants absolutely made all of this work possible. And then here's just another view of the field camp. So again, timber frames covered with tarps, and then hammocks suspended. You can kind of see them in the bottom right there. That was where we slept with our very important bug nets, and then we would have one tent that was dedicated to food and one tent that was dedicated to research. So that's where our microscopes and our collecting gear and all that was. Here's a little more of a close-up of the underside. This is in Cameroon. In Cameroon, we slept in tents on the ground, which was a really interesting experience for a number of reasons, because it was very wet and it flooded a lot. So as these trips progressed, my system of dugout trenches diverting water streams so that I wasn't sleeping in them just got better and better. And by week 4, I was usually like, okay, I'm not getting flooded anymore. So yeah, this is where a lot of the work occurred. And so the really fun part was that every day we would go out and collect mushrooms. So we would get out to our field site. It was quite a journey to get there in all cases, get our field camp set up, and then begin our actual research, which involved going out to one of 3 different plots and collecting mushrooms for about half the day, bringing those mushrooms back to our field camp and then analyzing them. We would sort of play triage, because you would come back with each day with a box that kind of looks like what you see here, right? There's all of these different amazing mushrooms, and some of them are more or less well known. Some of them have maybe been seen once or twice, but not for 100 years. And then some of them are just, well, these don't match anything in the known literature, so these might be new species. So very exciting to go out every day and see this incredible rainbow of diversity blooming on the forest floor. And then the slightly tragic part would be bringing it back to camp and being like, oh, well, that Amanita up there really needs to be worked on, and so does this Lepiota, and that means that we just got to leave the other ones behind, because mushrooms basically last for a day, maybe 2. in the condition where you can actually ascertain valuable scientific observations from them. So that's kind of a tricky part about mushroom collecting. You collect them, and then the clock starts. You know, you only have so much time to analyze the morphology of the mushroom before it starts to decompose. So yeah, I love this slide. It's so stunning out there. Yeah, so this is what I'm talking about. And this is us, this image is hiking through the swamp. So again, some of these trips were very, very wet, and the whole forest floor, or about half the forest floor anyway, would flood. And so trekking to our field sites was a very soggy affair. In the top right there, you can see me in my latter half of the day posture, where we'd have our mushrooms back at camp. And then I'm looking at, I'm measuring all of the features. You know, the pileus is so many centimeters across, so many centimeters tall. The gills are arranged in this way. The stipe is these dimensions, these textures on the surface. And yeah, and then again, in the bottom right there, I just want to point out that all of those collecting boxes full of mushrooms is from a single day of collecting, or a single half day of collecting, basically by maybe 8 people. So it's truly a rainbow out there. And so let me segue to Amanita. Amanita were, again, my mushroom of focus, and I feel very fortunate in that, because there are a lot of aspects of mycology that you might research, and when you tell people about it, their eyes kind of glaze over and they're like, what are you talking about? But if I'm like, okay, the emoji, right? The Mario mushroom, you know, that's what I'm talking about here. So it's, I think, the most easily recognized mushroom globally. Amanita are fascinating for a lot of reasons. They're very abundant, and they're very diverse. They're very colorful and charismatic. They have a lot of interesting tissues to look at, and they also, their shape corresponds to their relation to one another, which I'll talk about in a little bit. But you can see here an egg on the bottom kind of left of your screen. So this is what some Amanita will look like before they mature. They exist as these little eggs, and you can see the infant mushroom on the inside there. It has its mushroom shape, but it hasn't expanded yet. So that will expand to what you see on the bottom right there, and then that opens up, and the gills unfold and the veils break apart, and then they release their spores. So Amanita were my genus of study, and this is just a little breakdown of the fungal tree of life to show you where in the tree of fungal diversity we're talking about here. So we're talking about the Basidiomycota. That's the group that contains most of the things that people recognize as mushrooms. Mushrooms are fruiting bodies of fungi, which generally exist as a mycelium, a network of very thin filaments that has infiltrated a certain substrate, be that a rotting piece of wood or the soil on the forest floor. So those organisms that exist as networks of filaments will, at certain times, produce fruiting bodies, which we call mushrooms. And these mushrooms are means of sexual reproduction by which they can mix their genetic information with the genetic information of a mate and produce novel spores, which can drift off into the ecosystem and take hold and continue their pattern of life. So within Amanita is a very diverse genus. So Amanita includes, we think, at least 1,000 different species. So that red and white one, the fly agaric, that everyone recognizes so well, is only one of 1,000 species of Amanita. So it's the most easily recognized genus, I would say. But then when you talk about other species within that genus, I think oftentimes people have this almost synonymous understanding of that particular species, the fly agaric, and Amanita. But there are actually at least 1,000 different species of Amanita, and many of them are just as bright, colorful, big charismatic organisms as the fly agaric. So this is a little breakdown of the diversity within Amanita. So there are two subgenera, subgenus Lepidella and subgenus Amanita. So Amanita, the fly agaric, falls under genus Amanita, subgenus Amanita, section Amanita. So that's kind of how that works. And I'm just sort of listing these, there's not going to be a quiz at the end, so don't worry about memorizing them, but this will just sort of help reference where the Amanita that I found fall into the Amanita tree of life. So it's very diverse. I do want to say another reason why I feel extremely fortunate to have worked on Amanita is that by and large, you can look at an Amanita and you can say, okay, well, so it's got these dots on the surface. That means that its veil, its universal veil, is composed in a particular way. It doesn't have, or it does have, an annulus, a ring around the stipe. Okay, does it have a bulb at the base of the stem? You can collect all of these observations and you can assemble those observations into a picture that tells you, okay, well, this mushroom is most likely in Amanita, subgenus Lepidella, section Validie. Or this one down here, which is an Amanita from our field site, you would look at that and maybe some of you out there can look at that and say, well, what does that look like? Does anybody recognize that mushroom? You can mention it in the chat. But this looks kind of like what we might call a death cap, or one of the deadly Amanita, which fall under genus Amanita, subgenus Lepidella, section Phylogenetic, right? So you can look at this mushroom and say, well, that actually resembles the group of toxic Amanita. And that was one of the only cases was with these mushrooms where I would not attain the taste observation because I'm just not really willing to take that risk. By and large, when you're observing mushrooms, collecting them, you want to smell them, you want to look at them. I usually taste them because that can actually be an important identifying quality. And just taste it, chew it up with your front teeth on your tongue a little bit, and then spit it out. Yeah, make sure you spit it out, always spit it out. And again, with mushrooms that look like this, I'm gonna skip that step. But one thing you might not know about mushroom diversity is that in many groups of mushrooms, the physical appearance of the mushroom doesn't necessarily correlate so perfectly to the group that you're gonna find that mushroom in when you do DNA sequence work. So the DNA sequence work is generally considered to give a more accurate picture of the relationship between organisms. And so when you do that, when you collect those data and look at the results, you're going to see, oh, okay, so these mushrooms are related in this way, they're clustered in this group. That doesn't necessarily always correspond to mushrooms that look a lot like one another, or said another way, two mushrooms that resemble one another might be in very distantly related groups in the fungal tree of life. So all that is to say that Amanita is very, very friendly in the way that you can look at its physical appearance and figure out what group it's likely in within the genus. So Amanita are ectomycorrhizal mushrooms. That means they have relationships with the roots of host plants. And there's an image here that shows a schematic of the two most common types of mycorrhizal relationships. There's arbuscular on the right, which we won't talk about much, and ectomycorrhizae on the left here. So there are a number of differences between those two mycorrhizal types. I won't get into it too much, but Amanita are ectomycorrhizal. Most of the mushrooms that we were looking for were ectomycorrhizal mushroom species. And you can see here, these root tips are root tips that have been colonized with this fuzz here, this mycelium that's associating with their roots. So if you're interested in soil food webs, which I know you all are, I'm sure you've heard of mycorrhizae. I'm sure you are familiar with their vital importance to the health of all plant life. So with mushroom hunting, if you want to find the mushrooms, you have to find the plants that they're associated with, essentially. And so for ectomycorrhizal mushrooms, such as Amanita, chanterelles, porcini, or other boletes are also in this group of mushrooms that form ectomycorrhizal relationships with plants. So if you want to find those, you have to find the host plants. And here are some examples of ectomycorrhizal host plants. Depending on where you live, some of these might be more familiar to you. You're tuning in from all over the world. So us up here in my neck of the woods in Portland are probably going to be pretty familiar with the Pinaceae. That's, you know, firs, pines, spruce. The Fagaceae down here, those are your oak trees. Eucalyptus down here on the bottom right, if anyone's from Australia. But of course, eucalyptus grows all over the place now. And on the top left here, Fabaceae is the bean family. And there are many members of the bean family that exist as large tropical canopy trees. So within the Fabaceae, there's a subfamily called the Deuterioideae, and that includes the host organisms that we were searching for. So you can see this image here with this guy sitting in this tree. This is the tree called Decymbe corymbosa, and that was our host of choice that we were seeking in Guyana. And this is where our field site was. So yeah, so this was where we were flying out to. The interesting thing about these host trees is that if you're familiar with tropical forest dynamics, one of the things that is most immediately evident, especially for a person, you know, like me, coming from an ecosystem that's generally dominated just by a handful of different tree species. In the tropics, generally, you have a hyper diverse assemblage of trees that compose the forest structure. So you're not going to generally walk into a tropical or an intact old growth, especially tropical rainforest, and just encounter the same tree over and over again. You're going to see a lot of different trees composing the ecosystem. And yet we observe in a handful of little pockets across the globe in tropical locations, some species have developed an ability to carve out these vast swaths of land where they become essentially the only, or one of the only trees up to like 90% canopy dominance across entire landscapes. And so this tree, Decymbe corymbosa, you can see this old growth Decymbe tree here in the bottom middle. That tree is massive. The image next to it is a group of our team sitting inside the top up here. If you can, I think you can see my pointer. There's a little face right here poking out from the tree. So these trees are massive, and this tree in particular, I think, is one of the most amazing trees on the planet, not just because of the amazing abundance of mushrooms that it hosts, but because it grows in this Medusa-like fashion where it forms these adventitious roots that branch out and out and out and make these huge root mounds, and it forms adventitious shoots and has all of these many, many, many trunks. It's kind of a Medusa of a tree. And so this tree and our host tree that we were seeking in Cameroon, these trees both come to dominate the landscape of our field site, which is why we went there, because by and large, in tropical forests, you don't come across ectomycorrhizal host trees at this abundance. So in these particular field sites that are dominated by these ectomycorrhizal host trees, that is a place that you can go and expect to find a lot of ectomycorrhizal mushrooms, and indeed we did. So here's an image of where our field site was in Cameroon, in southern Cameroon. Yeah, so this tree is called Gilbertiodendron dewevrii. Our Guyanese host tree is also known as the Pallaway, so that one actually has a common name. This one has no common name that I'm aware of. There's not a whole lot of information out there about this tree, but it does host a lot of mushrooms. And I mentioned the biogeographic connection. So if you go deep into the Earth's history, you'll see that the continents that we see now were once mushed together in what we now call Gondwana lands. And as those continents split into the continents that we see today, the ecosystems that we can observe now developed, right, over a long period of evolution and various tectonic events. And so we observe, so our field sites, if you sort of take where we were at, one was about here in South America and the other was about here in Africa. So a long time ago, those continents were joined, and you could imagine that there was a continuous ecosystem that connected the two. And eventually these two continents split and developed into the ecosystems that we observe now. So the interesting thing about our research project is that we observed very similar and very unique patterns of diversity and ecosystem dynamics in our African field site as our South American field site. Those ecosystem dynamics are largely based on the canopy dominance of these particular trees, both of which occur in the Deuterioideae tribe of the bean family. So they're closely related tree species, and they host a similar diversity of mushrooms. So again, the ones that you're probably going to be familiar with: Amanita, chanterelles, there's clavulinas and boletes. So similar suites of mushrooms inhabiting these forests. And of course, that leads us to wonder, well, how did these forests develop in such a way? How did these mushrooms evolve? Did they exist before the continents split and then were sort of torn into two groups and then they developed in the way that we observe today? Or did the continents split apart and those ecosystems developed, say, maybe in Africa, and then there was a dispersal event across the forming sea? So that's sort of the macro, big picture question of the project. So again, one day of collecting, and yeah, those macro, those big sweeping questions that I was just talking about. The first step to answering those questions is knowing who exists where. So if you want to understand diversity, if you want to understand forest dynamics, first you have to understand who the players are. And so that's a big part of the research that we were doing, because in these particular ecosystems, there's very little information, in particular on the mushroom diversity. And of course, we think that the ectomycorrhizal habit is part of what confers this ability of these trees to dominate the canopy. And how is that occurring? We don't exactly know yet. But step one is, well, who's there? How diverse are they? Are these mushrooms in Africa actually closely related to those in Guyana? Or are they more distantly related? Are they part of some basal group of that particular branch that they share? So yeah, and that's where my work came in. So this, I'm not sure if I have credit here. This is not a photo I took. This is a photo that Noah Siegel took, who you might be familiar with. He's a prolific mycologist, mushroom photographer, and author, among other things. Also somewhat of a feral mycologist. He came with us and did some of our photography work. So yeah, thanks, Noah. And this slide is meant to capture the process of taking a physical mushroom that you find and turning it into words. So this is a picture on the right from my field notebook. You can see my really good handwriting. And this basically describes the exact shape, size, colors, dimensions of that particular mushroom. So as you can see, a lot can be said about a single mushroom. Not all mushrooms are going to require this much detail to describe, but Amanita have a lot of different tissues, and ones like this in particular that have these complex color patterns can require quite the verbose treatment. So here you can see a picture of our most familiar fly agaric, and these are the features that you're looking for to describe. So that description that you saw on the last slide is composed of basically this. It's usually a top-down approach. So you start at the pileus. You're going to say, okay, the pileus is how wide, how tall, what's the margin like? There these little ridges? Are there universal veil remnants on the top of the pileus? The universal veil is the tissue layer that encloses that baby Amanita. Amanita are unique in having that tissue layer. Most mushrooms don't. And then you move down the mushroom to the gills. You're looking at the gills. How are they spaced? How are they patterned? You're looking at the colors, the shapes. You're looking at the surface textures. Is it woolly? I mean, there's some of the mushrooms out there, and it's hard to capture this, but they would have a texture on the stipe that we would call cigarette ash, because it's so fine. It's like, you know, it's like ash on a cigarette that's about to fall off. It's that, you know, you breathe on it and it's gone. So sometimes it can be really hard to find a mushroom in the field and actually transport it back to camp without damaging it and damaging some of the important features that you'd want to capture. So this is the macroscopic treatment that each mushroom would get. And this is a time-consuming process. So again, this is why we would have, you know, this amazing box of diversity, and we've got a handful of people doing this descriptive work. I would manage, you know, maybe 3 or 4 mushrooms in an afternoon into evening until I was exhausted and starving and ready for dinner and to go to sleep. Yeah, but that it's a really important part of the process. And if you're out there and you're interested in doing this kind of work, all you really have to do is learn some basic botanical terms and descriptive practices, read some descriptions of mushrooms from other publications, maybe to use as a guide. That's what I did. Go out and collect a mushroom, identify it if you can. If you can't identify it, you can write a description like this. You can make formal collections yourself from wherever you are, and someone's going to want them. We generally want a better understanding of the mushroom diversity of our world. So wherever you are in the world, if you find something interesting, collect it, dry it, write down what it looks like, take some quality photos, bag up the little dried specimen, and get in touch with your local university and see if they're interested. Make sure you write down where it was, when, if you can, what kind of environment it was in. All of this information is important to our understanding of the health of our planet and our soil. So we would make these collections in the field. We would bring them back to camp and dry them using silica gel beads. So like the little packets that you get in your beef jerky or other food products with the little beads, right? You can fill a Tupperware with a thin layer of those beads and put a little piece of paper on top of that, cut your mushroom into kind of 4 quadrants, and then put it on the piece of paper, close up the container and wait about 12 hours, and the mushroom will be dry in the morning. So we would preserve our collections that way in the field, because of course we didn't have electricity or anything. And then we would take those collections back to our university in California, and then we would look at all of the different tissue layers under a microscope, which is the whole second half of the descriptive profile of a mushroom. So on the bottom left, you can kind of see a cross section of the gills. So one random skill that I have developed is the ability to take a tiny piece of a mushroom and a razor blade and shave off the thinnest little slice of a cross section. It's very difficult to get a section that's thin enough to see the elements without them all being just smushed together in this big soupy mess. So if you're trying to do that yourself and you're not so good at it at first, neither was I, and just keep trying, you'll get it. So these are all the different tissue layers, again, cross sections of a gill right here. This up here, the pileapellis is you can think of it as the skin on the surface of the cap of the mushroom. The cap is also known as the pileus. So these elements are the ones that are kind of sticking off. And it's cool because you can look at the tissue of a mushroom. So let's say the Amanita, the fly agaric mushroom, it has those white dots on the top of the cap, right? Well, those tissues are going to be formed more of these spherical cells that don't hold together very well. So it's kind of like a powdery tissue, right? That as the cap expands, those powdery patches get pulled apart into little powdery islands. So the cellular structure of that kind of powdery-like substance is going to be more like spherical cells, whereas if the universal veil is like a nice membranous cup at the base, it's probably going to be composed more of filaments, like you can see here, that are more tightly interwoven. So you can kind of guess what the microscopic appearance is going to be based on the macroscopic appearance. These watercolors that you see on this slide were done by a woman named Julie Henriette Martha Gusens Fontana, and she is my personal mycology hero. I definitely top of my list of people, if I could ever, have them over for dinner and have a conversation. I would love to talk to her because her work is almost the entire basis, the entire foundation of the literature that we were referencing for our Cameroon collections. She was doing mushroom collecting work in what was then the Belgian Congo in the 1920s, 19 teens and 20s. And so she was down there, she was out in the jungle, she was collecting mushrooms, and she didn't have a camera, so she made these beautiful watercolor illustrations, which actually, in my opinion, and if you're out there and you're an artist, do this. It might be kind of old-fashioned and photos can be really valuable, but in my opinion, her watercolor renderings of these mushrooms were and are more helpful than photos, because she can kind of capture the diverse range of features in a single image, whereas you often need multiple photos to do that. So yeah, her work is a lot of what we would reference. And this is another image of one of those amazing Medusa decymbi trees from South America. And these are a couple of our field assistants up in the top of it. By the way, no idea how old that tree is. It must be extremely ancient, but I'm not sure how you would possibly really measure the age of a tree like that. So yeah, the cool thing also about those trees is because they have such a kind of random, chaotic growth habit, every single one is utterly different. They're kind of like strangler figs. Some of you out there are probably familiar with strangler figs. They resemble strangler figs in a certain way, in the sense that the really old ones really start to resemble, I think of them as Ents from the Lord of the Rings, these massive old trees that are kind of taking a step across the landscape in slow motion. So yeah, so I'm going to move on to the mushrooms that I described for my thesis, and I'll just give you kind of a brief introduction to them. These are mushrooms that were described in a couple of publications that I made throughout the course of my graduate studies. And so without further ado, this is Amanita cyanochlorinosma. So the cool thing about, well, one of many cool things about this process was that I had the honor of formally applying a name to these organisms. And I'm a big fan of names that hold some kind of descriptive information, because if you look at the name Amanita cyanochlorinosma, you might be able to pick out a few things in there. Cyano, maybe think of cyanotypes, if you know what that is. Cyano refers to the color blue, essentially. So cyano refers to blue, chlorinosma refers to the chlorine odor that this mushroom has. This mushroom reeks of chlorine. It just has a very strong, it's kind of like you are, you know, in a over-chlorinated, you know, indoor swimming pool room. Very strong smell. And it has this beautiful steely blue-gray cap, so hence the name cyanochlorinosma. On the right here, you can see some line drawings that my friend Andy Goss produced for my first publication. So these are the elements that are generally considered important to describe in Amanita. So that's, if you look at the image here, A is the basidia, which are the cells that produce the spores. B are the spores. C are the elements at the tip of the gills, which is just kind of a special, unique Amanita thing to describe, as is D, which are the elements of the universal veil. So again, you can see that the elements of the universal veil are these interwoven filaments, which contributes to the texture that you see on this mushroom, having this membranous, sort of tightly woven universal veil at the base. So this one is in section Phyloides, which again, if you remember, is the same group that the deadly toxic Amanita fall into. So I think someone ventured to give this one a nickname and called it the something death cap. I don't remember, but I don't know if I like that name. So here is another one, Amanita fulvoalba. Fulvoalba means white and tan. So this mushroom just is kind of, this mushroom is really special in a lot of subtle ways that are kind of difficult to capture in a good name, I guess. But white and tan and Amanita in that particular part of the world, there's nothing else that fits that description. So that's why we settled on that name. It's something that you can, again, you can see that name and be like, you know, if you find a red mushroom, maybe that's not it. You know, I like names that can be helpful in that way. So yeah, again, the different elements. And I'll just kind of start moving through these and you can just look at them. This is Amanita guyanensis, named for the region of Guyana. It has a nice basal bulb here. This was one of the more common Amanita from our field site. You can see in B, it has these really nice spherical spores, which is kind of unique. So this is an example of one of our phylogenetic trees that we produced from our studies. So I think this was kind of a preliminary tree, so don't take it with, take this tree with a grain of salt. But basically what I'm trying to show here in this image is that when you're out doing this kind of work, you're making all these different collections. You're collecting one mushroom one day, one mushroom another day, mushroom another year. And you'll say, okay, I think these are all the same species. They don't match up with anything in the known literature, and I think they're all the same collection. And then you hope that you see something like this when you sequence their DNA. And you see, okay, yeah, all three of these distinct collections are have, you know, no branch length. That meaning that they are all very, very closely related, you know, at the species level, as opposed to, you know, this branch length, which is a little longer, which relates it to, you know, this other group here. So this is just to say we did a good job at identifying what we call morphospecies at first, you know, because before it's formally named a species, we can call it a morphospecies just as a way, and they have long names sometimes for our field work, like large gray annulate bulbous base kind of thing. And then we've used an acronym for that, right? And then you're like, oh yeah, MGAB or whatever. So yeah, moving on to Africa, this was, this image here is from one of our canoe ferries. We would have to take a little canoe route through a kind of swamp to get to our field site. This is, yeah, definitely one of my favorite mushrooms on the planet, Amanita Minima. This mushroom is only at most like 2 centimeters tall. It's a very, very tiny Amanita. I would say it is, as far as I know, the smallest Amanita in the world. And yeah, it's a really special Amanita in a lot of ways. I don't think I have time to get into exactly how cool this is, but you can read my paper if you want to. It's if you take a hand lens and you look at the surface of this mushroom, the particles, which you can see here, these spherical particles that compose the universal veil in figure C here, to the human eye, they look like little glass granules that are any color from the brightest yellow to the brightest orange to the brightest red. They're this amazing rainbow of sparkles covering the surface of this mushroom. It's just absolutely incredible. So that's Amanita, and Minima, of course, as you could probably guess, means tiny, right? Amanita luteolamellata, which is kind of fun to say. Try it if you haven't. Luteolamellata. That refers to, luteo means yellows, lamellata refers to the lamellae or gills of the mushroom. So yellow-gilled Amanita, which is very uncommon. Amanita, if you know how to identify Amanitas, one of the things that you'll know is they have white gills, almost always. But these do not. It's also very special. And again, I don't think I really have time to get into too much detail here, but for the group that it's in, which is the group on the bottom here, you can see section Vaginati. It is in the same group as grisettes, which you might recognize. And so being within that group and having a partial veil, this annulus, this ring on the stipe, is almost unheard of. So that's sort of a, like, wow, who's this new guy kind of mushroom thing, where you're like, wow, that is a very unique feature if you know your Amanita. So yeah, also, and that mushroom, by the way, can get up to, I don't know, maybe 1.5 feet tall. They're gigantic mushrooms. And so, yeah, I like to name things after features that you can observe, but like I mentioned, my ecologist hero, Madame Goussens Fontana, I just think that she deserves some recognition. And so this was one of the most spectacular mushrooms that we found out there, and I wanted to name it after her because I have so much respect for her and what she did. And her name isn't really in much of the literature. Her work, her collections were sent to a man in Belgium who published them and whose name is on the publications, right? So these mushrooms, most of these mushrooms that we worked on have the name Beely attached at the end of them. And they were all based on collections that she made. So I wanted to give her a little recognition here. This mushroom, Amanita Goussens Fontana, is named after her. Yeah, kind of makes me think of a jellyfish. It's one of these really, really fine powdery ones, again, that you breathe on slightly and the powder falls off of it. So real quick, the process of epitypification, don't worry about all these words too much, is where there's an organism that exists, it has a name, it's been found before, maybe it hasn't been found in a very long time. In these cases, since, it says here 1931 was when this mushroom was described. Conventions back then were not as advanced as they are today. I think people still thought mushrooms were plants back in the 30s. And so oftentimes there's these, and on the right here, you can see the original collection. This is that mushroom from 1931. This is that dried mushroom. For this process, which is basically updating the known information about an organism. So if it hasn't been found since then, right? Like, how are we going to know all the details of that mushroom based on our modern conventions? So yeah, on the right is that original collection. So when you're doing this work, you have to check the original collection, make sure it's the same species. And if it is, then you can add to the description. And so these are the following are mushrooms that I epitypified. So I did not name or describe these myself, but the modern understanding of those organisms is largely based on the work that we did out there. So this is Amanita robusta from that first picture. Really special mushroom, grows on dead termite mounds exclusively. So super interesting ecology there. This is Amanita bingensis, which is sort of the larger cousin of Amanita minima, who you met earlier. Again, it has that same rainbow sparkle to it, it's more of a sunset sparkle, I guess. But yeah, let's see, moving on, Amanita echinulata. I just want to point out real quick, if you look at the picture here, the photo, you can see that there's a little bend at the base of these mushrooms, a little torque to that basal bulb. And if you look at the watercolor image from Madame Goussen's Fontana, it captures that little torque of the basal bulb. And that's nowhere in the description, the original description of that mushroom, but it's captured in the watercolor image. And also, like, look at that watercolor image and look at the mushroom. That's clearly the same species, right? You almost just have to look at the picture and you're like, cool, we got it, you know? So again, she was an amazing artist and I have so much respect for her. So and this is just a really beautiful mushroom. And this one, Amanita fulvoalba, that's also a mouthful. Try saying that one 5 times fast. Fulvoalba. Again, you can see the photo and the watercolor image it's almost like the image, the watercolor is of that mushroom, right? It could be, but yet those mushrooms were found almost a century apart. So yeah, I'd say probably the most interesting, this is a really interesting mushroom for a lot of reasons too. This one also reeks of chlorine, like the whole field camp, right? Is just like, it smells like a swimming pool, right? It also has pinkish red mycelium, which is just a super interesting and unusual feature of a mushroom. So let's see, now I think that's it, that's the team. But in our Guyana field site, there are still 16 Amanita that we have recovered but don't know what they are. Cameroon has 47. And for reference to our field site, the total area is about a space in Cameroon of about 3 kilometers. So within a 3 kilometer area, there are 70 different species of Amanita, which just let that sink in, because that is astounding. That makes it one of, if not the most diverse location in the world that we know of in terms of Amanita. And Amanita, of course, is just one of many, right? So there's a rainbow of russula species. There's astounding diversity of bolete. There's all these other mushrooms, right? And yeah, so a lot of places are poorly explored, and so I encourage you to get out there. I just want to, that's pretty much, that wraps up that project. I want to mention something that I'm working on next, which is hopefully taking this kind of work to Madagascar. So we are trying to bring mushroom cultivation and taxonomic research to Madagascar, which is ecologically a place that really lacks understanding mycologically. And so we're hoping to fill some gaps there while also bringing education and seed planting and or seed exchange, tree planting, and mushroom cultivation as a means for empowerment to the Malagasy people. And we're hoping this can be a model that can be replicated worldwide, because I think it's a really powerful one. So if you're interested in this, please visit mobilelibraryproject.com. We need support in all kinds of ways. Please check it out. Thanks to everybody who helped me get here. And I'll leave it at that. So feel free to keep in touch. I invite anybody here to send me an email, follow me on Instagram, visit my website. Yeah, and some latest offerings down there. And then here's just some sort of collage of extra photos to look at. And with that, I think we'll move on to Q&A.
Oh, thanks so much, Kennan, for bringing this to us today. And man, you got a lot of fans in chat, a lot of people. You know, I said I feel like a kid in a candy store looking at all these photos with you. So yeah, I will share out if you'll stop sharing screen for just a minute. I hope
Absolutely.
people got a chance to see your contact information, but I will go back to sharing we're all the way to Q&A section, like we said. But you know, one thing I do want to pause just a moment, if I may, and point out something for those who come to our webinars that are really like more soil nerds than mycology nerds. I'm always looking for things that we can see with our eyes that help us understand what we should imagine the soil is like. And the fact that you've gone into these places and found such diversity, like relatively unknown in modern science diversity, is really telling, because the knowledge of humans about soil, the soil story and all its complexity, you know, it'd be a lot to estimate it at 5% at this point in history. And so this is just one more example of where if you go to any of those marginal spaces, any of those slightly like under-resourced research areas, you will find, it'll just smack you in the face how diverse this planet is. And you know, some estimates say like that there's maybe 13 million fungal species out there, and modern science knows 150, 200,000 maybe. So it's just really exciting to me, and I wanted to thank you personally for teaching so much to us today.
So welcome.
Yeah. Elaine or Laura, did you have anything else that you wanted to say before we jump into questions?
Nope.
Yes, sorry.
Go ahead.
I just want to comment how important it is, like doing this taxonomic job and research like Kennan did, because it was also very complete. It includes like morphological characteristics, ecological characteristics, but also the molecular analysis, which is also a great tool. But I believe that maybe all this information could be relevant. It depends on what you're looking, but they complement each other. So it gives you like a more robust hypothesis, because at the end, giving the name of a fungi and a classification is an hypothesis. So the more information you get, the better. So I think that this kind of work is really, really important. It's like to open a door to a new world, unknown world about this fungi, and is making these organisms visible also for people of the community. So it's been great to see that you work also with people from the community, and also it has a lot of relevance for example, for legal protection, or to look at the below ground protection. And actually, I wanted to mention something about it, because I know that some mycologists and experts are looking for an equivalent term for flora and fauna. So they proposed the fungi. Have you heard about this?
Yeah, I've heard of that. I've also heard mycota.
Mycota. Oh, cool. Yeah, I like the F because it's the 3 Fs. So yeah. So
yeah, it makes sense. I'm on board.
That's awesome. Well,
Thank you.
You know, you brought up, Kennan, about some of the ectomycorrhizal relationships that are common in Amanita species. And so we did have some questions where I saw in chat too that people get really excited, like that mushroom is the tip of the iceberg of what's going on below ground, right? And so I was wondering, like, I mean, we'll open this up to anybody who wants to mention this, but we got a couple questions here about this concept of the wood wide web, right? This is like Suzanne Simard's work, many other people, that is there this common mycorrhizal network that's going on out there? And this person is saying, you know, what do you think of that? And that it's very popular in temperate regions, but what is it happening in the tropics? And do you think that the fungi are facilitating things like plant communication in these tropical settings?
That's a really good question. It's a really interesting and complex question. And I think, like any interesting and complex question, it has a very nuanced answer that we probably only vaguely understand currently. There's some really interesting research that has demonstrated this kind of thing. And actually one of the other graduate students in my cohort was doing a project that was trying to answer a question right along these lines. So let me try and summarize this really quick. There's this there's a theory of plant dispersal that it is of the benefit of the plant to get its progeny dispersed as far away from the host plant as possible. It's the Jansen-Connell dispersal hypothesis, I think is what it's called, something along those lines. Anyway, generally true, right? That's why you make a plant that looks, or that's why you make a fruit that looks like something that an animal wants to eat, and then it's going to poop out elsewhere, because the idea is that you don't want the progeny competing with the parent, and also there's going to probably be some shared pathogens that are already in place on the host that might get spread to the progeny. So that is, I think, a pretty well accepted model for a lot of ecosystems and organisms. And the other graduate student that was looking at this ecosystem from more of that perspective was actually trying to make an argument for the opposite in this particular instance with these particular trees, that they actually have she was trying to demonstrate some degree of shared mycorrhizal network by which the adult plant would confer a competitive advantage to their progeny via below ground networks of some kind, probably mycelium, because they all share these mycorrhizal connections. And so the idea is basically that by operating in that way, they're able to take over these huge swaths of forest that come to dominate entire landscapes and tropical ecosystems, because the mature resource-rich trees are providing extra, sugar to the baby trees down in the forest floor that aren't getting all of the light access, and then those trees are able to grow bigger to eventually take the place of their parents, and the ecosystem expands. And any other, would-be canopy tree that gets its start there as a seedling is going to get outshaded by those trees that have the advantage. So I think that there is a strong possibility that the wood wide web is occurring on some level in these ecosystems. I think that when you're talking about tropical forests at large that aren't composed of single tree species like these particular ecosystems where we worked, I think you're dealing with a much higher diversity of plants and a different, not necessarily overlapping system of arbuscular mycorrhizal fungal associates. And I honestly don't know anything about how and if those fungal communities are connecting to one another and communicating and delivering resources and communications and whatnot. But I think it is likely occurring on some level here in these ecosystems.
There's a fair amount of work that's been done here at Oregon State, where we were looking at those kinds of questions. We found that there were mycorrhizal mats in the soils around any of the babies that would protect that root system, that the mycorrhizal fungus was connected into mama tree that might have only been 3 feet or 6 feet away, and absolutely not enough sunlight getting to the surface of the forest. So how could that baby plant that could have been 3 years old or 10 years old or 20 years old, it still looked like a little baby plant, yep, but it was alive and functioning. And if the parent tree got pulled over for some reason, like, some of the root system was infected, or it just happened to be in the wrong place at the wrong time, and when really bad weather came through and was, knocked over, then all the baby plants, all the little guys, grew more rapidly than any of the weeds or other plants. And you were left basically with a forest of young trees that were perfectly capable of duking it out with other organisms. there's the work by Ted St. John, that worked down in Brazil along the Amazon. And he showed that there really was no difference in the number of species in a wide number of places up and down the Amazon. So that to me says, I don't think there's any real difference between temperate and tropical systems. You just have very, very different species because temperature and moisture are going to be limiting, or they're going to be overwhelming. One, you know, it's one end or the other. You're either in the middle of a drought or you're in the middle of a flood. And you have to have the mechanisms for dealing with a completely waterlogged soil versus just the water you may have been able to save in a aggregate status. The aggregates built by the bacteria and the fungi will hold 50% of the weight of that aggregate will actually end up being in the water that it's holding, and then the roots of the plants can come down and get to that source of water late in a very dry period of time so that they make it through the drought part of the cycle. So I think mycorrhizal fungi can do a lot of things. They're there. They've been doing these things for a very long time. We just didn't register them in any way or any shape that these were important parts of the ecosystem. So get to work, students. You've got a lot to do.
Well, I really like that you brought up, Kennan, the ecosystem differences when you have such a diversity of tropical plants that are probably associating with arbuscular mycorrhizae, because, you know, that's kind of where I've done most of my research on arbuscular mycorrhizal fungi in grasslands. And so typically grasslands may have 16 species per meter square, right? And it's interesting the way that you can have competitive advantage based on the association. If certain plants like late successional grasses, for example, can really they can get so much benefit from the mycorrhizal fungi that they outcompete other plants. You can also see times where the mycorrhizal connection between plants seems to facilitate coexistence there. But then you're saying in your system, it's more homogeneous naturally with the species of trees. And so I think that there is even more opportunity for, they don't have to kind of shake hands at arm's length. They're like, we're sort of the same thing in an evolutionary sense. Let's cooperate more. And I think that that's probably really a factor. It's where the tools are still just not there sometimes. And I do want to address the other question about the critical paper regarding the Wood Wide Web. I've read that paper. I really respect the people that wrote it. I think basically they're saying, you know, has this ever really been measured? And so it's a little bit more of an academic question or a fight between scientists about whether or not the conclusions are outpacing the methodologies that we have to work with. Obviously something is happening. Like maybe it's not a direct connection. Maybe it's that the organisms are spilling different resources into the soil matrix, and then they're facilitating each other or signaling, you know, some way. But it gets into a little bit of, I think, a reductive conversation to go into the conclusions of that paper too deeply, because what we're still doing is everyone is trying to grapple with the unknowns. And it's worth criticizing if the conclusions are getting ahead of our methods, but it's also worth us reaching for creative ideas, because nature's full of examples of where, you know, parasitic plants connect to trees through fungi and get carbon, those kind of things. So it's at least plausible, in my opinion. But I don't know, does anybody have a follow-up on that?
I guess the one thought that comes to mind is that I think it's easy to look at an ecosystem such as a tropical ecosystem, such as the one I worked in, and say, oh, well, okay, it looks like these trees are, kind of coming to dominate this forest. It's like they're trying to push everybody out. And I think, at the risk of personifying them too much, I wonder how much the plants are controlling those dynamics over large scales of time and how much it's the fungi. And the fungi are saying, hey, this is really good habitat for us and all of our friends. Let's see, how much we can expand this if we just team up with this really powerful ally. Yeah.
Because it is the plant that's controlling the exudates that are being produced that the fungi would have to have while they're trying to track their way to the root and get something established. So you don't often see that. Here in the Pacific Northwest, we have some pretty nasty tree diseases. And usually when the plant becomes mycorrhizal colonized, the disease-causing fungus disappears. So I think there's something going on. How specific is that interaction? Because I am thinking of just one fungal disease-causing fungus example here. So, have I gone through and assayed all of the disease-causing fungi in this part of the world? No, not by a long shot. No. I had enough to do with the soil food web. Didn't need to add all these, and that's why I always come to these kinds of talks, is to expand my knowledge and make things, we've got to tie it all together. What are all the interrelationships, positive and or negative?
Yeah, one other thing real quick I want to point out is that, I talked about mushroom hunting a lot, and that's kind of how we're getting to know who is there and whatnot. It's really important to know that mushroom occurrence does not necessarily give you an accurate representation of what's going on below ground. And that is a really difficult thing to get a picture of. It can tell you, if you find a mushroom, then you know that organism is there, right? That mycelium is there. It might be a very minor part of that, of the complex below ground dynamics that include many other fungal species that aren't ever going to give you a clue that they're there. So it's a complex picture, and part of the research that we were doing, other than the mushroom stuff, was doing some root excavations and finding ectomycorrhizal root tips, putting those into morphological categories, and then sequencing those to try and get a better, more accurate picture of what's actually going on below ground and how frequent each different mushroom group is actually found on the roots when they're sampled.
That's really neat.
Because it changes with season too.
Yeah.
So it's a great thing to keep you occupied for the rest of your life.
It would not be a wasted career. I think we better move on because we have several more questions in about half an hour here. So
sure. Just
in the interest of time, what are some of the relationships you observed between the Amanita species and local plants and animals in the locations that you were in?
That's a good question. So as far as plants go, I think that's a pretty simple answer, because those Amanita will be found nearby an ectomycorrhizal host tree. And in the Guyana site, that was pretty much just Dicyme corymbosa. So that's the main association with Amanita with plants. In Cameroon, there were a handful of other ectomycorrhizal host trees. So there was a Wapaka species that grew kind of down in the more swampy parts of the forest, and then the Gilbertiodendron trees that grew in the less swampy parts of the forest. And certain mushrooms would associate with would regularly be found next to one or the other. Sometimes there was overlap, and it was, you're like, well, it's kind of in between a Wapaka and a Gilbertiodendron. But for example, if you think back to those two mushrooms that looked very similar to one another that I showed you, Amanita Minima and Amanita Bingensis, the sunset sparkly ones. Super similar mushrooms, right? Well, one is regularly larger and associates with Gilbertiodendron trees. The other one is regularly smaller, very diminutive, and associates with the Wapaka trees. So, so the association with a particular host, in that case, becomes an important part of separating two similar species from one another. So that was a big part of the argument that I used to say that they are in fact two different species, because Amanita bingensis had already been found, known, described before. That was just epitypified. Amanita minima, I found, and at first I said that this is Amanita bingensis. It's clearly, very similar, but the sizes didn't match up, and the host didn't match up, and the habit didn't match up. It grows in these little troops, whereas the bigger one only grows in singularity. So, so yeah, the association with one plant versus another can be an important taxonomic feature, discerning one species from another. And then as far as animals, there is, in Guyana, there is one Amanita species, I forget the epithet, but the Patamona call it Pulutukwe. And that mushroom is a choice edible that is recognized by, we didn't really talk about, I didn't really have so much time to talk about the mycophagy habits of the Baca and the Patamona, which I think are really interesting topics. I feel like through all of this work, I've, part of me is like, oh, do I want to be a biologist, or do I want to be an anthropologist, because there's just some really interesting stuff going on down there. I mean, they recognize the mushrooms. So the Baca in Cameroon, their language captures the ecology of the mushrooms that they recognize. So certain mushrooms, and they're largely thinking about the mushrooms that they can eat or do something with, or that are a marker of some kind. But the name in their language for a particular mushroom, let's say that grows with the Gilbertiodendron tree, is the name means, mushroom Gilbertiodendron, kind of, or like mushroom Wapaka, or mushroom, a different species of tree. So I think it's really interesting that their language, those people, the Baca, have inhabited that region of the world, the sub-Saharan lowland tropical rainforest wilderness of Africa, for no one's actually really sure how long, many thousands of years. It's thought perhaps as many as 10,000 years, if not more. So they are a people whose entire way of life and entire religion is the forest. They are the people of the forest. The forest is, it's kind of a deity. It's a plural deity that guides them and takes care of them, and that they interact with and understand on a very deep level, which is why I always try and be as careful as I can to say that I for these mushroom species that I applied a Latin name to, right? I described these species. I introduced them to the scientific community. I did not discover them, right? All of these mushrooms that are out there are known to the people who live there, and they have a relationship with them oftentimes. And so, yeah, I think it's really important to acknowledge that. And yeah. So, and then the other thing is, as far as animal fungus interactions go, the big one is insects. Flies, fly larvae are the bane of my existence. Yeah, I'll leave it at that. You harvest, you pick the mushroom, you're like, I'm going to describe this. You don't get around to it that night. The next morning, it's soggy, stinky mush, because the maggots are prolific in the tropics.
You didn't happen to bring your refrigerator with you?
No, we'll be excavating a root cellar next time.
Yeah. That's a good point. Yep.
Well, I love the concept that you brought to the table with that story or that explanation, which is, you know, we're operating in a language here on this webinar that doesn't have a lot of embedded ecology in it. We don't describe things by their relationships, right? We describe an object, and it's one of the reasons why it's a hard language. It doesn't make any sense that English is the language of the international world communications, except for, you know, some historic colonial stuff there. But, you know, what you're saying is that people that are living so in touch with nature, which is something that we really think is critical for humanity, for the rest of us humans to evolve towards, is to live closer in relationship with nature. Part of the whole shtick for regenerative agriculture and natural farming and the things that we talk about. We have to rediscover how to know things by their ecology, by their relationships, how to have those conversations. And I think it's really neat to think they describe the mushroom by the tree that it grows near. It's an amazing concept.
Yeah, I agree. I think this makes me think of a way that I frame things to, like, friends, family, or just people that I talk to, because I meet people and they're like, oh, what do you do? And I'm like, well, where do I start? I think that most people have this kind of what's known as a green blur when they go out into the forest, and it's just kind of like, it's a green blur. And I like to, like, what are all these things? I like to compare that to, you know, imagine you go to a big party and it's someone's birthday party, and everybody there, all these friends who've been best friends for 10 years, and you're the only one who doesn't know anybody, and you kind of walk in the door and you're like, okay, I feel like I'm just maybe going to go drink some punch in the corner and then maybe leave early, you know. Whereas if you go to a party and you walk in the door and people are like, oh, and you see a bunch of your friends and they're like, hey, come over here, we're having a conversation, or, you know, I liken that to when you go out into your local ecosystem and you have learned at least a handful of names, you know, and not even names, but just identities. You don't even have to know what things are called. That's just a tool that humans use to approximate the truth, which is just what exists out there. So if you recognize the face and you think, I know that plant, that's that plant that grows next to that mushroom, and, you know, then you gain friends. And then when you go out into the forest, it's like you're showing up at the party hosted by a bunch of friends instead of a bunch of strangers.
I love it. Yeah, that's great. Laura, did you have anything that you'd like to add, or shall we move on?
Yes, I just wanted to add, for example, here in Costa Rica, we have lost a lot this connection with, for example, feeding on fungi. This is something that I know that, for example, in the United States is very common, or in Mexico, Oaxaca, that you can collect fungi from the forest and they know what to do with this fungi. And this is something that we lost. And so I was wondering if you think like having a diversity is also a challenging, or let's say like fungus have this stigma sometimes about, because they're related to decay, to death. So I was wondering if it is challenging in a diverse place to identify these different species. So it would be more difficult for people to connect with those species than in other, I don't know, areas where you already know like the 5 species and the one that is toxic or something like that. So what do you think about that?
Yeah, that's a really interesting question. And it makes me wonder, because in both of these cultures that we worked closely with, the Baka and the Patamona, they both exist in this ancestral wisdom framework by which they know that this mushroom is edible, and it's basically because that's what our parents ate, right? You know, and all sorts of amazing. I mean, oh my gosh, there's this, how they know to do what they do, and to how they know all of the ways that they can interact with their landscape. In Guyana, one day they went out and harvested these lianas, this particular kind of liana that had this milky white sap. And then they went and found a little narrow tributary creek, and they took these bats and they would mash up these lianas and swish them around in the water. Meanwhile, another couple of guys are making these nets on poles, like lacrosse sticks, kind of. So they're mashing these lianas, swishing them around in the water, and the water's getting kind of milky. And that milky sap stuns all of the fish. There's all of these little, like maybe up to a foot long catfish and other little fish. And all of those fish, they just get stunned and kind of float up to the surface. And then they would walk down these little tributaries and just scoop up the fish in their nets.
Wow.
How they figured out how they can do that is, an ancient mystery. And I think on the one hand, it's hard to say because the mushroom diversity is so high there, and yet they know some mushrooms that they can eat, and they largely avoid eating most of them. I will say there is one really interesting occurrence of, I don't even know what to call it. Essentially, when my advisor, Terry, started doing these trips down there, so he was, just for a little bit of background too, he was a collector for the Smithsonian, and he was doing some plant collecting back in, I think, the early 90s in the Guyana interior, using these Patamona folks as guides, back when, this was before a lot of these folks were even hunting with rifles. They were using bows and arrows and slingshots, essentially. And so he's down there, they're guiding him around, and this is where he stumbled upon this ecosystem where, all of a sudden, wait, all of the trees are the same and there's mushrooms everywhere, and there's nothing in the scientific literature about this. And so that he catapulted his career into what it has been. And so once he started studying the mushrooms down there in Guyana, one of the mushrooms that he came across is a Craterellus species, which is the species or the genus that includes black trumpets or yellowfoot chanterelles, and a variety of other edible mushrooms. It's a genus which, in which all known species are edible, right? And they're commonly occurred up here in North America. It's one of my favorite edible mushrooms, black trumpet. And so there's this Craterellus species that Terry's finding down there in the habitat where the Patamona live. And Terry knows that it's an edible genus, and so he's observed that these guys, they eat. There's this Amanita that I mentioned that they like to eat. There's a Clavulina species that they like to eat, which is just amazing, nutty, delicious mushroom. And then there's this Craterellus species, and Terry's going, well, do you eat that mushroom? I mean, if it's not edible, it's the first known inedible Craterellus. And they're like, no, we don't eat that one, And there's not much to be said for why. It's just that's not what their parents and grandparents and ancestors did. They just, they didn't eat that mushroom, and so they still don't eat that mushroom. Well, in a very bold. So Terry described, this was a new mushroom species that Terry's team formally described some number of years ago. And Terry took the very bold move of sampling it. This mushroom that he had a good reason to believe was edible, but absolutely no evidence of it being edible, because not only was it an undescribed species, but the people who have existed with it since time immemorial did not eat it. And of course, the Patamona are just kind of excited, like, that one's not edible. What do you do? But he started eating it and sampling it cautiously at first, right? As you should do. By the way, any edible mushroom, even if it's a known edible mushroom, if you've never tried it before, just eat a little bit of it, because people can have sensitivities even to known edible species of mushrooms. Just throwing that out there. So he started sampling it. It tastes really good, and no one's getting sick. They start eating more of it. It keeps tasting really good, and no one is getting sick. And after a time, the Patamona, they're observing this, and at first they're just like, no thanks, but then they're like, okay, I guess we'll try it. And now they eat it. Now it's in their mycophagus habit. Now they have added a locally abundant mushroom species to the mix. And I just think I think that's a really fascinating example of, I'm not even sure what to call it. It's almost like scientific wisdom supplementing ancestral wisdom in a way that I don't think I've ever heard of happening before. So yeah, interesting story. Thanks for asking.
That's really neat. The way you even just said that, it's like I'm trying to embrace spaces where science can bring something really critical to the table and to the discussion without overpowering indigenous knowledge, philosophy, the experience of farmers or land managers. You know, and it can be very complementary, and everybody can gain from the exchange if we tread lightly.
Yeah.
I think science is often used as a bludgeon, but it doesn't have to be. So let's move on because this, I think, maybe a quick answer, but you know, some curiosity about your genome sequencing work and what you did in the field versus maybe with a collection that you took back to a lab.
Yeah, that's a good question. I meant to mention this earlier, but I didn't. One of the critical steps in mushroom collecting for our project, and I would encourage anyone to find a way to do this if you're interested in doing this kind of work. When you collect the mushroom and you bring it back to where you're going to look at it before you do anything else, and write down all of the details of how it looks macroscopically, and then you're going to dry it somehow to make your formal collection, and then you can analyze the dry tissue. If you have a microscope next to your fresh mushroom, like, cool, look at the fresh tissue. That's way better than drying it and then rehydrating it, which doesn't always work great. Anyway, before you dry it, you can take, and this is what we did, take a little sample, I would take a razor blade and I would cut out a tiny piece from the center of the mushroom, kind of like the top middle of the stipe, like where the stipe and the pileus meet. Cut out a little piece of that tissue and drop it into a tiny little vial that had some, it was called C-tab, I think. It's basically a buffer. It's a buffer solution that keeps the DNA stable until we can get it to a lab where we can extract and amplify it. And so, due to the constraints of A, we had so many collections to work with, and B, sequencing costs money. We did kind of a broad scattershot approach of just trying to get a handful of genes from each of our collections of interest sequenced. So if you look at the paper that I published, we were using, I think, 4 different loci. So there's the ITS, the internal transcribed spacer, which is sort of it's like the fungal barcode, sort of, or traditionally it has been, I think, and I've been doing other stuff for a couple years, so I'm probably more out of touch than some of you are with this kind of stuff. But there's different sequences from the genome that get pulled out and analyzed, sequenced, and then compared with other collections. And that's what we use to make our phylogenetic trees. Different fungal groups are going to have other more specific genes that can be used that work particularly well in that group. Like, for example, it's going to be a different set of genes in Amanita versus Boletus, or something along those lines. So we were only sequencing, I think, 4 genes for all of the collections that I worked on. As far as genome sequencing, if you're talking about actual full genome sequencing, hasn't been done to my knowledge for any of our collections. And I know that it's just getting easier and cheaper and faster to do all that stuff. So if anyone out there is interested in sequencing, I've had some people approach me and ask about the different Amanita toxins and things that are produced in Amanita species, and if they could use some of my collections to see what levels of those compounds are produced in these rare tropical fungi versus the ones that we're more familiar with. There's all sorts of other genes that we didn't sequence that could be sequenced. All of my collections, all of all of our collections exist in bags, in boxes, in a fungarium somewhere. Mostly in the United States, but also it's worth mentioning that part of the agreement that you make with a country when you do this work is that the original voucher specimen, the epitype, the collection which represents the sort of the model of what that organism is supposed to be, that will end up back in the country of origin. So, you know, our collections are, or those type collections at least, were kind of on loan, essentially. So we're going to work on them, you know, maybe describe a species, and then that original collection ends up back at the fungarium in Cameroon or Guyana. So a lot of work to be done still in that regard.
Yeah, this question is good. It also just opens a whole can of worms, because I know, you know, I've dabbled a little bit in the taxa identification of arbuscular mycorrhizal fungi, and it's like you can literally watch scientists at conferences yelling at each other over, no, that's not right, you're not supposed to use ITS for that, it doesn't work well, you're overestimating diversity, you're underestimating diversity, well, my database is the right one that everybody should be using. And it just turns into it's like, it's just science is
it gets
messy.
heated. I've seen it happen. You're right.
It's like, children, calm down.
It sure can be. You know, it's important to remember scientists are just humans, right? So, well, that's great. I think that I hope that helps give a little bit more context there. What about spores?
Yeah.
Spores help you identify species.
Absolutely. Yeah. And I didn't bring too much attention to it, but if you were to look back at my slides, you would see that I've made an illustration for each one of these species that I worked on. And so each species has a particular spore shape and dimension. So some of them are sort of, I mean, some spores are kind of cylindrical, some are more, and there's words for all of them. There's lemoniform, which means lemon shaped, fusiform, meaning kind of tapered at one end, piriform, meaning pear shaped. And so this is, you know, part of the vocabulary that learns into, or that is required to do this descriptive work. But essentially, yeah, they're all different. Interestingly, within the genus Amanita, as I talked about sort of towards the beginning, there's the two subgenera, right? Lepidella and Amanita. So all Amanita fall within one of those two groups. The way that you differentiate between those two groups is that that subgenus Lepidella has spores which turn blue in an iodine solution. And subgenus Amanita has spores which do not turn blue in an iodine solution. So spores are a really important diagnostic quality for mushrooms, but they range a lot too. So it's important. So a lot of the work that I actually did in grad school at home in the lab was under a microscope with a little micrometer in the eyepiece of the microscope, measuring one spore at a time and getting the dimensions. And then I get this wide range of dimensions. And then I'm kind of like, okay, most of the spores fall within this range of sizes and shapes, with some outliers, ranging out to here. Also, interestingly, there's a guy, if you're really interested in Amanita, go to amanitaceae.org. That's the family name of the Amanita family, amanitaceae.org. It is this curated website developed by mostly a guy named Rod Talas, who is, in my opinion, the foremost Amanita expert in the world. He's a really incredible guy. I think I don't think he has any institutional affiliation. I think he just kind of does his own thing. Super fascinating guy. Almost all known information about all Amanita is on that website and organized in a pretty accessible way. So all of the information from my publications is on there already. And he has, I know that he has a software that he plugs into his microscope that sort of automatically measures the spores. So like cuts down a lot of the work necessary. So that kind of software is out there and makes this work a lot easier. But yeah, check out that website. It's really amazing. Lots of big, beautiful pictures. Yeah.
That's awesome. Yeah, you just gave me a flashback to a conversation I had with Joe Morton, who ran the International Collection of Mycorrhizal, Arbuscular Mycorrhizal taxa for a long time. INVAM it was called. And he's like, I can barely go to like poster sessions with students, posters at conferences because I see too many students saying, here's a picture of my spores, and I'm like, those are nematode eggs or something, right? It's really hard to sort those things out, and people actually used to have to break the spore and then try to see the layers to differentiate the taxa. And now with molecular work, we don't have to do that as much anymore. But just a fascinating whole discussion. Elaine or Laura, what have you all found related to spores here at the school? Because, or beforehand, Elaine, as the school is not the only part of your scientific career.
Yeah. Well, when we're looking at the soil food web, we want to talk about the active, living, doing something, function is happening. And spores don't do much other than just sit there and wait until the right conditions come along for them to germinate and start to grow. So we've kind of, because it is so very hard to tell differences in the different types of spores. It just got to be too difficult to try to put any information in how many spores did you count, because remember that you can have one conidium or one I love it when my brain just goes bleh. It's conidia and the other one is sporangium, and they're going to produce thousands of spores, and so you see one spore in your soil suspension. That doesn't mean very much. And then you've got to try to figure out what was that spore from. And there's just too many things that have spores that look alike. Sorry.
I have, my experience is related to entomopathogenic fungi. I work a little bit with those, and it was something challenging because there are a lot of cryptic species, so they look exactly the same. It's not possible to differentiate the species by the morphology, but they have difference, for example, in the type of host that they prefer, or in some aspects about their biology. So that's when molecular biology is a great tool to help to understand also like cryptic species. Mm-hmm. And I was wondering if there is any like cryptic species for Amanita.
The answer is almost certainly yes. So I can tell you, taxonomists are generally considered to be either lumpers or splitters. You may have heard that term.
I think the crew has heard that before.
Yeah.
I'm a lumper.
So, okay, I think I'm a little bit of both, because when it comes to the actual formalization, I'm a lumper, because I'm more along the lines of, well, what is actually useful to a person out in the field, right? But in the field, I'm definitely a splitter, because I want to capture as much information as I possibly can. And so if two mushrooms, and so for the Amanita, in Cameroon in particular, the Amanita within section vaginati, which are defined by a handful of qualities, like they have a membranous universal veil cup at the base of the stipe. They usually have grooves in the margin of their cap, and they don't have an annulus, except for the ones that we found. But they also, so let me just say that slide towards the end where I said we have 70 different morphospecies of Amanita, that in our Cameroon field site, that is a splitter perspective. That is taking every little difference and saying, no, that's a different morphospecies and that's a different morphospecies, when a more accurate representation of the relationship between these mushrooms might just be that the exact color on the surface of the cap, presence or the, the texture of the universal veil, the presence or absence of an annulus, these things might all be kind of plastic phenotypes that are different expressions of a sort of more homogeneous genotype pool, where you could make an argument that they're all kind of a species or a species complex. And yeah, I think that if you were to take every single little variation on a theme of section vaginati from our field site and make each one of those a different species, it would be an extremely cumbersome fungi.
I just shudder to think about doing that, every single species out there, and you've got every tiny little difference could mean something. I want to wait until you show me that little tiny difference actually makes a difference.
Yes.
Otherwise, they just go together as one species.
Exactly. Yeah.
That's a really great tension to have in the room between the people that say, well, what's the functional, practical difference? And then other people to say, well, like, let's look at this again, because maybe we can find out that something new about the story of maybe they do have enough slightly different genetics, and maybe there is a reason, and that we can learn something from the evolution there. But those conversations are going to go on and on and on until we figure it all out. And I don't predict we're going to figure it all out until the year 3055.
And I think something that I want to bring attention to that Laura mentioned earlier that I think is really, really important to keep in mind for all of these conversations is that species is just a hypothesis.
Exactly.
What we call a species is just our human approximation of the reality of that is occurring in the natural world. And so I think maybe we can have some humility about our observational capacity as the beings that we are.
Absolutely. Well, I know we're a little past time, so I do want to acknowledge that we had a whole support team with us here today. Sammy and David, Meg, Alex, you've all been involved in this behind the scenes, helping with sorting out questions and everything. So thank you so much. And to you, our panelists, for being here. Kennan, for you to share this knowledge with us. And I, by the way, dropped a few links to some of your papers in the chat. Okay. So maybe your H index is going to go up over time as people cite you now. And yeah, this has just been so much fun for me. I think we've had a lot of people stick around all the way to the end. So I think we're going to get some great feedback on this one.
And thank you very much for hosting this and keeping us on track and moving on to the next question. Thank you so much, Adam, for being here as well.
Well, appreciate that. And I think with that, Kennan, I'm going to send you an email because when I come to Portland next time, we should hang out.
Absolutely.
Continue the conversation. So,
and thanks for having me, everybody. This has been a real pleasure. And please, anyone get in touch anytime for any reason. And thanks everybody for tuning in.
And when we get our new office in McMinnville built, and we've got a lab, and we hope to be doing DNA work there, stop by and get the tour.
I certainly will.
Great.
Thank you.
Fantastic. Okay.
Cheers, everybody.
Right.
Ciao.
Bye, everybody.
Thank you. Bye.
Don't forget to click that like button.